Okay, so let's continue on. This is one we've had. We know the answer to this is polyp. All right, our next question. Identify the term that is used to describe the evolutionary history of a particular species. I'm not going to give you one of these, but if you look over to the right, you have your ancestral protist at the bottom because we came after the animal kingdom came after Protista. And then we have multicellularity that splits, that's unique, not unique, but is common to all. Now, to the left are sponges. They don't have true tissues. Everything after the sponge will have tissues, although the cnidarians only have two layers, ectoderm and endoderm, from the flatworms up to chordata, the chordates, which is what we are. We all have the three layers, endoderm, ectoderm, and mesoderm. So what you're looking at actually is not genealogy or biology or paleontology. This is what's called a phylogenetic tree. Again, it just shows you where everything splits off and where your common ancestors are. So phylogeny is your answer. All right, distinguish between asymmetry, radial symmetry, bilateral symmetry, and give an example of each. All right, well, you should know what asymmetrical means. That means no symmetry. Radial means that we have a central axis with appendages radiating out. And then, of course, bilateral means that if a plane goes from head to toe, each side is a mirror image. So here's your answer. Asymmetrical, there is no symmetry or balance in the body shape or structure, and there is no shape. So your example would be porifera, which is the sponge. It does not have a shape at all, so they're asymmetrical. Radial symmetry, the animal's body can be divided along a plane through a central axis into equal halves. This just means that we have a central unit with the radiating parts. Think of your jellyfish, think of your starfish, that's radial. And bilateral, the animal can be divided into mirror image halves along one plane. Anything after, except for the starfish, anything after your jellyfish is going to have bilateral symmetry. All of your worms do, your dogs do, you do. Everything has got bilateral after the cnidarian, with the exception of our starfish. Okay, explain why it would be beneficial for a fish to lay many, many eggs when fertilization is external. Now, this means that the fish has laid eggs, the female, on a reef or at the bottom of the ocean or water, and then the male fish swims over it and deposits the sperm. So why do you think it would be beneficial for the female to lay a lot of eggs when that's the type of fertilization they undergo? I think we all know the answer to this one. Some of the eggs are going to float away. Some of them are going to be eaten or destroyed. So you have to, it's like, why is there more sperm than eggs? So this is to ensure fertilization. A lot of those eggs are going to be damaged or they're going to be eaten by another fish. Just think of Nemo. Remember, she laid a whole bunch of eggs, and then that barracuda came along and ate up all but Nemo. So a lot of things happen when there is external fertilization. All right, which is not an animal characteristics that define all animals? Well, we know, starting at the bottom, that all animals are multicellular. We know that they're all heterotrophic. They have to eat other organisms in order to get their energy. And they're all eukaryotic. We all have a nucleus and membrane-bound organelles. We all have mitochondria and ER. We have all of that in our cells. But the one thing that all animals are not, that is going to be carnivores. We have a lot of herbivores. Think of cows, they're herbivores. We have insectivores. We do have carnivores. we have omnivores. So not all animals are going to be carnivores. All right, at which stage is the developing animal an embryo? This is a really good question. Which one of these? Now we have the egg and the sperm here. See the little sperm and the great big egg. And then this is your zygote. Your zygote happens right after the egg and the sperm meet. And then it starts dividing like crazy. And then when we get over here, we have our blastula. Our blastula is this hollow ball. It's got fluid in there. This is going to eventually be ectoderm. And then our next stage is the gastrula. The opening is the blastopore. This space is the archenteron. The archenteron becomes the gut. Everything we see in blue here is ectoderm. Everything we see in tan or pink here is going to become the gut and the lining of the gut. So at what stage do you think you have an embryo? Because we talk a lot about embryos, but is it 2 divisions? Is it C? Is it not an embryo until we get to D? Or is it well, this one's not a choice. Gastral is not. So is it A? Is that an embryo? I don't think so, because you don't have the joining of the egg and the sperm yet. Here you do, and here you do. So at what point? B. Now, this goes to everything you've been taught. I mean, especially through religion, that when the egg and the sperm meet, we have an embryo. An embryo is a developing zygote. So the embryo is the stage at which the zygote forms, and then it's going to go through its multiple divisions. All right, which reproduces by internal fertilization? Okay, so we haven't been over the fertilization of all these things, but just think about it. We have a clam, we have a fish, we have a bear, and we have a frog. So which one do you think reproduces by internal fertilization? I think the obvious is the bear. All right, what is the most recent evolutionary development exhibited by both arthropods and echinoderms? Now, we really haven't talked about arthropods and echinoderms, but arthropods are going to be they have crayfish, there's lobster in there, octopuses in there. Octopus is not in there. We have insects in there, so those are our arthropods. Our echinoderms are going to be our starfish and our sea urchins. And we know that as adults, they have radial symmetry, but they're deuterostomes, which means they are the phyla right before us. We're both developing that anal opening first. We're not protostomes. So when you look at the two, arthropods are protostomes. Echinoderms are not. And us, which are chordates, we're not. So the only two phyla out of all of them that are deuterostomes are your echinoderms and us, which is chordata. So if you look at the answers, there's a body cavity, a coelom, which is the same as the body cavity, segmentation, which we know that allows us to be flexible, or protostome development. The actual answer here is coelom. The reason for this is because echinoderms are radially symmetrical, and so that implies that there is no head, there is also no cavity. Now, this is not a question I would ask you because this is a little bit further advanced and we really have not talked about our echinoderms. All right, which organisms are most closely related to roundworms? Well, when we look at our animals, we have sponges that have nothing. We have our cnidarians. Cnidarians are radially symmetrical. Flatworms are our next phylum, and then we have our arthropods. Our roundworms are going to be between C and D. So our next chapter is going to be on flatworms and roundworms. Arthropods are the ones most closely related because flatworms they are considered to be acoelomates. They're flat. They don't have a coelom. But roundworms do, and so do arthropods.